Nā mihi nui kia koutou katoa. Nau mai haere mai, piki mai, o te welcome to Rangahau Rising webinar. You are in the right place. Ko Nick Waipara tēnei. I'm Nick Waipara. I'm going to be facilitating, emceeing today's proceedings. So thank you for coming, everybody. We really welcome your attendance. But before we get on to the real meaty stuff, i.e. Alexa, I'm going to open today with a karakia timatanga. Kia ora te marino, kia whakapapa o ngā mo. Te moana, hei huarahi mā te tau, e te rangi nei. Aroha atu, araha mai, tātou e tātou e. Homi e, hui e, tae ki e. Ti hei mauri ora. Welcome, no mai haere mai. Thank you. Before I introduce Alexa, just some of housekeeping. Please look at BA's socials, the Insta, all the socials are there, and you'll be seeing them in the chat. So you can engage, you can see what events, what to register on. You can actually look at our amazing Ngā Mihi Kia Korua, Hana the previous Rangahau Rising presentations. If you missed their kōrero, their talks, you can still see them online. And also you can engage with BA really interactively. So I urge you to do that. Secondly, I'd like to give a mihi, ngā mihi kia koe huata for who's ko Pou Whiranaki of BA, and his mahi around celebrating the Te Wiki o Te Reo Māori, Ake Language Forever. It's the 50th anniversary of Te Wiki o Te Reo Māori, or Māori Language Week, which runs from the 14th to the 20th of September. And a shout out to Huata, who's been doing E Mohi o Rānei Koe, or Did You Know, and on a daily, and also the kupu of Te kupu o te rā, or word of the week, and ngā mihi, because today's kupu is Mānuka. So I thank you for that whakaaro and that kaupapa that's going out to you all on the BA mailing list. And with that, he's provided also a whakatauki. So also to open Alexa's presentation today, I've not used Huata's whakatauki. I've done one especially for Alexa and the kaupapa, which a whakatauki is a proverb or an aspiration, a saying that represents a whole lot of values or reo or around topics or subjects. So my one and my kupu for today is oni, or soil, and the whakatauki today is te toto o te tangata he kai, te oranga o te tangata he whenua he oni. So that really talks about the blood and the health of ourselves and feeding and nurturing ourselves, but it is the land and the soil that's really underpins all that. So without further ado, I would like to open today's Rangahau Rising with the amazing Dr. Alexa Byers, and her kōrero today is studying drivers of soil carbon resilience across agroecosystems in Waitaha, or Canterbury. Alexa is an early career researcher specialising in microbial ecology, and her research is to understand how the environmental disturbances impact on oni, or soil ecosystem health. She's undertaken a PhD, an amazing PhD, I must say, at Lincoln University in 2021, and now is working and worked as a soil carbon ecologist at Scion, now the BSI, before joining Bioprotection Aotearoa as a postdoctoral researcher, originally from Cumbria, which is in the north of England, correct, north of UK. That's where she hails from, but we just see her as Alexa from Aotearoa now. So no mai haere mai, welcome Alexa. Please introduce yourself and what you would like to say before embarking on your kōrero. Kia ora.
Thanks for the introduction, Nick. I have a pepeha to give, so I'll just share my screen. So, ko Scafell Pike te Maunga, ko Wasdale te Roto, ko Cumbrian te Iwi, ko England a Ho, ko Christchurch o e Noho Ana, ko Alexa toku inoa. So thanks for all coming. That was just my pepe ha, just to give background where I'm from, and I'm currently based in Christchurch.
Kia ora. Thank you for that. And we can see your screen sharing, just so you know.
Okay, great. Thank you. So kia ora koutou. Thanks for being here today. I'm really excited to share one of the research projects that I'm working on at Bioprotection Aotearoa. The focus of this project is exploring how we can sustainably manage and potentially maybe even enhance soil carbon storage in Canterbury's agroecosystems down here in the South Island. So to kick things off, I'll give you an overview of the wider project and what we hope to achieve over the next few years. And then I'll touch on why soil carbon matters and why it's so relevant to bioprotection specifically. And then finally, I'll walk you through some of the findings from a study that I carried out in Canterbury as part of my postdoctoral research, which really lays the groundwork for where this project is heading over the next few years. So our project sits within the research pillar of BA called Po Nuku Arangi. The big picture goal of our project is to understand the factors governing the long-term stabilisation and persistence of soil carbon across agroecosystems. And of course, I'm just one small part of a much bigger team. And I'm really fortunate to be working with Dr. Nicola Day from Victoria University, Leo Condren from Lincoln University, and Shenjing Shi from Ag Research. And we've just had two fantastic PhD students join us a few months ago as well, who are going to be doing a lot of the hard graft for us. So we're very lucky to have them. So over the next few years, our research is really centered on three key themes. So firstly, we really want to understand how land use change, and modification affects the long-term stability of soil carbon. To do this, we'll be using a range of kind of biological and chemical methods to figure out not just how much carbon is in the soil, but how stabilized it is and how resistant to degradation and loss it is. And a well-known example of a common land use change in New Zealand that I'm sure you're all aware of is afforestation of grassland, that is planting trees, whether with natives or non-natives, which is incentivised for a variety of reasons, to offset carbon emissions, diversify revenue streams for farmers, and increase carbon sequestration across agroecosystems. So just using this example, understanding how afforestation does impact long-term carbon stabilisation, not just kind of storage, is important to assess its efficacy and also, most importantly, where it can be implemented across the landscape to have the greatest benefits for everyone. So secondly, this one's a bit more mechanistic. We're really interested in studying the underlying mechanisms that drive the fate of soil carbon from different kind of management strategies. So how do things like soil biotic and abiotic factors like soil organisms, the underlying chemistry and the physical conditions decide whether soil carbon inputs are stabilised in the long term or just lost back into the atmosphere? And to get at this, we'll use slightly more experimental approaches like cover cropping trials or kind of drought experiments, like a range of different experimental approaches. And then third, we're exploring how stabilized soil carbon changes with depth. So soil carbon dynamics below the topsoil, so below kind of 30 centimeters, it's still a bit of a mystery. There's more and more work happening, but most research has been conducted in topsoils, as that's where most of the plant and microbial activity occurs. However, deep soils generally thought to be more stable and longer lasting, so understanding factors that can support the buildup of deep soil reserves in agricultural landscapes will be really important for securing long-term carbon storage. So if we're interested in bioprotection, then why study soil carbon? Soil carbon can seem like a bit of an abstract concept. When you think of bioprotection, you commonly think of protecting kind of flora and fauna from pests and pathogens and diseases. However, although those things are very important in their own right, soil carbon protection is highly relevant to human environmental health. Simply put, soil health is fundamental for maintaining agricultural productivity. For thousands of years, we have depended on soils for the provision of food and raw materials. And more recently, in the past few decades, we've begun to recognise that soils have a natural capital and they provide a range of services that sustain human health. So we depend on them for not just food production, but also regulating air and water quality and playing a role in greenhouse gas regulation. However, with agricultural intensification and large-scale changes in land use, we're seeing major shifts in the biodiversity, functioning and health of soil ecosystems, which threatens the capacity of soils to continue supporting these essential ecosystem services. So, we need to manage soil health so that we can still kind of get all of these amazing things from the landscape that we depend upon, but also we can kind of sustain and maybe even enhance the services that it provides in the long term. Things that, the services that underpin soil fertility, crop performance, and agricultural productivity. So, in simple terms, soil health is, the cornerstone of protecting socioeconomic and food security in Aotearoa, New Zealand. So again, why focus on soil carbon if we're interested in soil health? Well, the amount of carbon stored in soils is actually a really important measure of soil health. The quantity, quality, and cycling of soil carbon gives us a really strong indicator of both soil quality and soil productivity. On top of that, soils play a massive role in the global carbon cycle, and they store more than 80% of the Earth's terrestrial carbon stocks, which is actually more than the atmosphere and the plants combined, which makes them, critical for long-term carbon storage. And if we manage soil carbon carefully, soils can act as a long-term carbon sink, which is a really powerful tool for reducing CO2 emissions and helping tackle climate change. So knowing all of this and knowing that we want to protect soils, but we also depend on them in right here and now, if we want to develop sustainable land use practices which meet today's needs, whilst also protecting soil health and productivity for the future, we really need to understand how historical and current land use and management is affecting soil carbon storage. So just one last step on this winding path. So if we're interested in soil carbon, then why study the soil microbiome? Well, to really understand soil carbon cycling, we also need to look closely at the soil microorganisms. Why? Because soil microorganisms exert a really strong influence on the breakdown and processing of soil carbon. The kind of diversity and the metabolic activity of the soil microbiome are key factors that determine soil carbon turnover and processing, and then the consequent release of CO2 into the atmosphere and that loss of soil carbon. What's more, when land use and management practices change, the kind of ecophysiological traits of soil microorganisms can shift, which can have direct consequences on the flux of soil carbon and the long-term persistence of the soil carbon pool. And, interestingly, compared to plants and some soil abiotic properties, microbes respond quite quickly to environmental change and stress, which makes them a really sensitive bioindicator and, essentially an early warning system for monitoring soil health and ecosystem functioning in response to change. So now that I've kind of set the background as to why soil carbon is so important for bioprotection, I'd just like to share some insights from a research project that I carried out at Bioprotection Aotearoa during my postdoctoral studies. So this was undertaken over a couple of years, and I carried out a research study which aimed to examine the impacts of land use and management on soil carbon and microbial cycling dynamics across an agricultural landscape. And I was lucky enough to be working down in Kaituna and Priest's Valley in Banks Peninsula in Canterbury, which for anyone familiar with the Banks Peninsula region, they sit just below the slopes of Mount Herbert and Mount Bradley. And these valleys were ideal because they contain a really diverse mosaic of land uses, each with different histories and management practices. And that kind of land use diversity makes them a great representation of the kinds of land uses on agroecosystems that we see not only across Banks Peninsula, but also in New Zealand more broadly. So these are just some images of the beautiful landscape I was working in, and they kind of show how over quite a small area you get a really diverse range of land uses, from irrigated and dryland grazed pasture, regenerating scrub, some native forest fragments, and Pinus radiata plantations. And they were very scattered across a relatively small area, but had very contrasting land use and management histories, which made them a really good study, kind of with relatively controlled environmental conditions across the landscape. So now the part we all love, methods, which I won't spend too much time on. So I sampled soils from 5 different land uses across the valleys, which included areas of native forest, which were remnant stands of old growth podocarp forest with many native tree species. Areas of regenerating scrub, which were formerly, pretty unproductive grazed pasture sites that are now regenerating with plant species such as carnuka. Areas of planted exotic forest, which kind of like the regenerating scrub sites were formerly areas of unproductive pasture, which were converted into Pinus radiata plantation. Dryland pasture, which was kind of non-irrigated sheep and dairy pasture. And then the 5th was irrigated sheep and dairy pasture. And the image on the slide shows an example of what these sites looked like. I think you can see pretty much all 5 land uses in that one image. But we did sample more replicates of those land uses across the landscape. We weren't just limited to what's shown in that image. So to understand not just the soil carbon, but also microbial carbon cycling dynamics in each land use, we carried out a range of measurements. So first we did basic chemical analyses, which included total carbon and nitrogen, labile carbon, bioavailable nitrogen, pH, phosphorus, and exchangeable cations. Next, we measured basal microbial respiration to estimate kind of the baseline microbial activity. And then we also use phospholipid fatty acid analysis, or PLFA, to estimate microbial biomass and then the relative proportions of different microbial groups like fungi, bacteria, actinomycetes, gram-negative and gram-positive bacteria. And then finally, we examined microbial function and the contribution of microbes to soil biogeochemical cycling. So for this, we performed metagenomic sequencing on soil DNA extracts. And then using various bioinformatic tools, we annotated microbial genes involved in soil carbon and nitrogen cycling, which allowed us to see how microbial functional activity varied across the different land uses. So, what were the key findings? Well, as always, what seems to happen with my research, I did not find what I expected to find. So overall, even though there were, big differences in land cover and management regimes across the landscape, the differences in total soil carbon concentration between the 5 land uses is actually quite small. Both dryland and irrigated pastures had the lowest total soil carbon concentration, and the biggest contrast in carbon was between the regenerating scrub and the dryland pasture. I just want to really importantly note here though, this is just total carbon concentration. We didn't take bulk density measurements, so we didn't calculate stock. So this just reflects concentration. And if you look at the error bars and all the plots, you can see there's a lot of variation in soil carbon, even within each land use type, not even across the land use types. So, soil carbon really just vary a lot across the landscape, regardless of land use, which is important to think about when we're evaluating the effects of land use. Similar patterns were observed for labile carbon, which is it's with hot water extractable carbon. It's kind of a measure of the readily available carbon, how accessible it is to be eaten up by microbes. This displayed relatively minor differences across the sites, and there was also a large variation even within each land use type. The dryland pasture and irrigated pasture sites did also have a really low soil C/N ratio, which is probably quite unsurprising. There were no large differences in total nitrogen between the sites, so the low C/N ratio in pasture soils probably is due to the slightly lower soil carbon content in these soils, not necessarily shifts in nitrogen. And yeah, just something to consider as well. We primarily looked at total soil carbon. We did not assess kind of the chemical stability or the recalcitrance of the carbon. Having these metrics would provide us a really more detailed understanding of how resistant this carbon is to degradation and breakdown. And that's something that in the next few years we're really going to be tackling, really looking at ways to measure the kind of the resistance of the soil carbon in the soil. So looking at the results of our microbial function analysis, we found that more intensive pasture land uses had a higher richness and diversity of microbial carbon degrading genes, particularly compared to native forests. We did see a clear distinction between the kind of modified and like pastures or exotic forest or whatever, and the unmodified land uses, which suggests it's not just the type of land use, but it was also just land use change in itself was associated with an increased abundance of microbial carbon degrading genes. Similar to the patterns in total nitrogen, we didn't really see any differences in the diversity of microbial nitrogen cycling genes, which kind of tells us that land use seems to have a more selective effect on microbial carbon cycling than nitrogen cycling. So the patterns we saw in the diversity were also reflected in the abundance of microbial carbon degrading genes. Native forest soils had the lowest abundance compared to the 4 modified land uses, which again, it just appears that land use change in itself stimulated an increase in microbial carbon degrading genes. So why might this be happening? Well, one explanation may come from the intermediate disturbance hypothesis, which just kind of postulates that kind of disturbance may stimulate increases in microbial diversity as microbes adapt to the new environmental conditions, which in this example would be kind of different carbon sources and different inputs. And previous studies have shown that kind of increasing land use intensity, like turning diverse natural ecosystems into agricultural monocultures, can see an increase in soil microbial diversity. In more intensive agricultural land uses, you might see a higher diversity and abundance of microbial communities as the kind of soil microbiome adapts metabolically to the changes in plant communities and environmental stress. When the land use changes, the plant community diversity shifts, and that might open up niches for different microbes to occupy and gives a reduced advantage to specialist microbes that kind of thrived under those stable native conditions. And also kind of favour microbes that have a more flexible genomic range to metabolize different compounds. Yeah. So interestingly, even though the native forests had the lowest diversity of microbial carbon degrading genes, they actually had the highest microbial biomass. So in other words, there were plenty of microbes in the soil, but they just had a lower functional diversity and a lower genomic potential to break down soil carbon. We also found that fungal biomass and also the ratio of fungal to bacterial biomass was highest in regenerating bush and exotic forest. This probably reflects the fact that these soils have quite low soil moisture conditions, as bacteria are generally considered more sensitive to dry soil than fungi. So to wrap up, we didn't really see any major differences in the total soil carbon concentration between land uses, although the pasture sites tended to have the lowest carbon concentration. Land use change was linked to increases in both the diversity and the abundance of microbial carbon degrading genes, and we saw a clear distinction between modified and unmodified land uses. with the native forests showing the lowest diversity and abundance of these genes. So overall, our result, based on the microbial kind of functional data, these results suggest that, kind of large-scale changes in land use may increase the functional potential of the soil microbiome to break down carbon. However, it's important to note that this hasn't really translated into changes in the actual total soil carbon content yet in these landscapes, but it might be something worth factoring in the future. So, this would have important implications for how land use and management kind of influence long-term carbon sequestration in these agroecosystems. And just one final point, future research is needed to understand the chemical stability of the soil carbon pool across this landscape in a much more detailed way than analysing kind of total soil carbon concentration. And there's always plenty of people to thank. So I'd like to thank my postdoc supervisory team during this period, which was Dr. Steve Wakelin from Scion, Professor Amanda Black from BA, and Professor Leo Condren from Lincoln. I'd really like to thank Williston Farms and Matt Monger for allowing us to sample the soils at Katuna Valley and Priest's Valley and being so helpful and welcoming. I'd like to thank the New Zealand eScience infrastructure for providing the computational resources for the metagenomics analysis, and this funding came from a TechCore grant to bioprotection Aotearoa.
Oh my te paki, virtual clap for Alexa and your here. He kōrero pai. Great presentation, great talk. I didn't explain, but the wonderful Zohar and Anna have explained in the chat. There are no live questions, but you can ask a question in the chat, which I will relay and filter to Alexa. So please, he pātai, any questions? Another kupu or word, pātai, questions. So please go ahead and any questions to that amazing research. I was interested in the fact that you'd found that stability, that stability in the native forest ecosystem. That's quite, well, I guess it gives some surety, some, I guess, comfort is the word, or some that you are seeing that, and why the importance of native soil ecosystems is really important to actually enhance that stability. And also, would you think also re-inoculate with the microbiomes or some of the
Yeah.
beneficial or those carbon sequestrating microbes that may come from that area, that fragment, into, let's say, horticulture, agricultural areas that might be losing some of that biodiversity. I don't know. Do you see that?
Yeah, I mean, it definitely highlights the value of having native fragments on farms and, working really hard to protect those. Not even just for soil carbon storage, but just for how beautiful they are and how important they are to kind of our, cultural heritage of New Zealand. In terms of inoculating microbes, I guess that would depend on whether the microbes are responding to the above ground conditions, kind of that the lack of disturbance and the kind of plant communities, or whether those are kind of inherent properties of the microbes themselves. I would assume that they're responding to the above ground conditions, perhaps.
Yeah.
So it would be interesting to see what would happen if you inoculated them into a different land use, whether they would transfer those properties or whether they would kind of adapt to the new conditions and change. Yeah, it'd definitely be an interesting area of research.
Cool. Kia ora. Ngā mihi Dan Tompkins, kia ora, welcome. Lovely to see you here. Dan has a question for you. Outside the current focus, but any thoughts on how browsers, domestic and or wild, might be influencing soil carbon? Good question.
Yeah, again, that's a really good question. And I think particularly in terms of disturbance, and particularly it's all connected, right? Those above ground communities are feeding into the below ground communities. So once, if you started to see large shifts in plant biomass and a kind of reduction, maybe either an increase or reduction in below ground carbon inputs from browsers, I think you would expect to see a shift in how much carbon is stored. and particularly if the soil profile's being disturbed and kind of the conditions are changing, um, that would definitely be something to consider.
Yeah, particularly, terms like peak cow and, the pest imbalances with those vertebrate pests. But yeah.
Mm. I think
Uh,
soil
oh,
carbon generally takes a little bit longer to respond to all of these changes. I think there has been some research done which has looked at the impact of um, non-native browsing and kind of predators in kind of nature reserves and looked at, please correct me if I'm wrong, anyone in the comments, but I think that, they didn't necessarily see massive changes in soil carbon with kind of disturbance, but I partly think that's just because it takes a long time for soil carbon to respond to changes. It can often take, a decade. Um, there's a bit of a lag effect in soil carbon.
yeah, so that's interesting. Um, Kia ora, Zoha. How much is plant cover and diversity of the plant cover, let's say, do you think is driving what you're seeing?
Yeah, that's a really interesting question because I was very surprised. I thought that there was going to be more diverse microbial communities in the native forest soils because they had more diverse plant communities. I thought it was going to kind of mirror, and I think that's generally what the science kind of assumes or hypothesizes. This almost went completely against that. I kind of saw a higher diversity in land uses with a really low plant diversity. So I guess there are many other environmental factors and also kind of human anthropogenic factors which are kind of influencing how the microbes are responding and kind of shifting microbial diversity. I think plant cover will is a really important factor. Um, but there's also going to be, a lot of influences from management and disturbance that will kind of affect microbial diversity too.
Kia ora. Kia ora, Edward. Alexa, you referenced the C to N ratio degrading microbial ratio. Do these different microbe types compete so as C degrading microbes increase, say, nitrogen cycling microbes decrease, and vice versa, and I guess that those sorts of parameters.
Yeah, that's a good question. Um, yeah, I definitely did see a much more selective effect of land use on carbon degrading microbes versus nitrogen. The nitrogen cycling dynamics of the microbes weren't really affected that much. There was no real differences amongst the land uses. Um, whereas, the large differences were in kind of the abundance of carbon degrading genes. Um, whether they come, sorry, I'm struggling to answer this question. I guess I can't necessarily determine from this data whether they compete. There were definitely shifts in the communities, but I guess because the nitrogen cycling genes of the microbes weren't necessarily affected, it's kind of hard to determine whether they were competing or not, or whether carbon cycling was just more affected by land use.
Yeah, that's interesting. And I guess if you have any afterthoughts, you can still add to his answer in the chat or later, because that is quite a hard question. And then you've got other drivers in place, don't you, as well?
Yeah, yeah.
Um, Kia ora, Gabriel. Thank you for your question too. And I've just scrolled off it. So, did you compare the soil carbon of hill country versus the valley floor soils? Any differences across water and dry soils? So, you
Yeah, we tried to avoid going too high up the valley sides because we kind of sampled in the valley and the regenerating bush and the exotic forest soils. They were kind of on slightly elevated areas which used to be pasture but were kind of taken out of pasture land use because they just weren't as good quality soils. Um, but we did try and limit the elevation because we didn't want that to become kind of a huge influencing factor. Um, Soil moisture was definitely, I didn't really talk about it in this presentation because the focus was on carbon. The soil moisture was definitely something which was influencing the data a lot too. Um, I think I mentioned that the fungal to bacterial ratio was much higher in drier soils. So there was a big shift in dynamics there, which did really mirror soil moisture conditions with a kind of increase in fungi in the kind of more drought conditions, I guess you would say.
Mm. It was quite interesting that your results, you have been surprised by some of them so far. As you say,
Yeah,
future research is still required.
yeah. Because
But,
even, I mean, talking about the fungal to bacterial ratio, there was actually a really low fungal to bacterial ratio in the native forest, which I assumed would have the highest fungal to bacterial ratio, but not, yeah, it didn't. Yeah.
well, at least that, yeah, and it'd be interesting to see why. I mean, or so what's driving that, and added to that, can you what can you speculate um, about resilience and land use changes and multiple changes over time, i.e., native to pasture to forest? So I guess, can you speculate what you're seeing with those opposite results?
I think the this research highlights the value of protecting and having native forest fragments on farms. When you're looking at it at the landscape level, when you're looking at the ability of the landscape to sequester more carbon, as opposed to just focusing on one land use type, it does really highlight the value of having that kind of diverse range of land uses across the landscape, um, and kind of, yeah, but kind of protecting native forest fragments, because they were kind of associated with the lowest abundance of carbon degrading genes. Um, Obviously we can't convert everything to native. That's just not, we need to use our land to produce food and eat and all of these amazing things. Um, and I guess kind of having that balance across the landscape of native, like protected native areas, and then, productive pasture land uses.
But it also, as you say, there's that tension between loss of, let's say, grazing pasture and pasture productivity for livestock productivity.
Mm,
And that is some concerns that some of the farming sector have raised,
yeah,
and
yeah.
horticulture sector, primary production sector. And yes, you can restore fragments, let's say for catchment restoration and other kaupapa like that are important. But I guess this research is really pointing to other, as you said, the other um, ecosystem services. I hate that German word.
Yeah,
What I
yeah, for
mean,
sure.
and it gives it gives the sectors like other reasons besides. Let's say you're nowhere near a catchment, like you can still be doing retiring areas for fragments of a
Yeah, like
vegetation
perhaps
to
unproductive
get these
areas,
sort of
or
factors, right?
yeah, 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 for sure. And again, like, I think I focus on soil carbon. Soil carbon's really important. Um, but sorry, I've just realized, am I still sharing my screen, or can you see me?
Yes, we can see you,
Oh,
and
right,
we've got a
sorry.
lovely, we have a
I just had a
lovely,
moment where I was
and
like, did
there's
I stop
actually a
sharing my
lovely
screen?
vista as well.
Um, what was I saying? Oh yeah, soil carbon's really important. But I also think when you're dealing with land uses, I'm always cautious about turning it into a ranking system of like this or that,
Yeah,
because there's many other values you get from different land uses. You get amazing values from pasture, from like timber production, and you get amazing
yeah.
values from, no, sorry, you get amazing values from pine, like timber production and things. You get amazing values from pasture. And also with native forests, we do have that deep connection with the native forests, and everyone loves walking through a beautiful native forest. Um, and I guess it's a balance of like quantifying soil carbon and assessing that, but also assessing the other values of the land uses and having a balanced approach to it all.
Hmm. He Pata, any questions, keep putting them in the chat, but welcome to have any more questions. But I'm coming back to my favorite rant, well, one of my favorite rants is about country calendar and fantastically promoting I guess, the innovation in primary production and I guess the whole policy around regenerative agriculture that sort of came in the previous government, but I'm not entirely sure where that is now with that policy. But I guess some of what your research, the longer term application of it is to influence, I think, in my opinion, and you can debunk me if you're wrong, if you think I'm wrong, is to influence those sort of policies around both climate change mitigation, but also what we're doing in our primary production, first, like in terms of sustainability and soil health. And given another little factoid or e mohio rana koe. Did you know, and most of you here will know, that New Zealand's one of the worst places on earth for losing its productive topsoil through
Mm.
just plain old erosion and mismanagement, or
Yeah.
let's say less than ideal management. And so soil restoration and valuing soil and that topsoil, I think everyone assumes that everyone's a country calendar farmer, and I don't think they are. And and it's a learning journey for them. So the sort of knowledge that you and your team and others at Bioprotection Aotearoa and beyond are producing, I think is actually going to be pivotal in changing that behavior or attitude that soil is some sort of infinite
Mm.
resource that we have in
Yeah.
Aotearoa. When in fact you look at the Manaaki Whenua Landcare researchers' data around the sedimentation and what's happening to our topsoils, it's absolutely it's grotesque is a word, I guess.
Mhm.
But do you ever see, for example, your research leading to more than just applying policy or applying pressure to those who aren't country calendar farmers, is to like an app or a soil tool that farmers themselves or growers themselves or communities, or kaitiaki, people that actually are working in long-term, crop production could be using to actually monitor their own soil carbon, what they're doing, how they're improving or not improving, Do you see
Mhm.
it being rather than sending a soil sample to a lab, like Hills, for example, do you ever see it being what you're doing eventually going to be doing directly informing the farmer? Rather than it being filtered through federated farmers or a lab or a CRI or whatever. Sorry, that's a long
Um,
question, but
I think there's already quite a lot of work being done in setting up those kind of platforms. I guess the issue might be that soil's actually a really complicated medium to study. I don't mean that in kind of a airy-fairy, like, oh, I'm a scientist kind of thing. It's hard to find a single measure of soil health. Um, it's quite complicated, and it would be finding a measure that people could reliably use that was consistent over many different land use types and for many different purposes, um, that isn't just kind of selling snake oil, I guess, of like,
yeah,
this is a secret to a healthy soil. That would be what I would be
Bi-
cautious of. But
biochar.
yeah.
Sorry, snake oil. Um, no, and also I guess that you mentioned the people you're working with, like, and the questions come from kia ora. I'm just have to scroll up to see who you are. Apologies for that. Around the community itself. I'll just have you had any feedback from the community about your work? Also, what are you looking forward to most with the next phase of your research, like the future research you
Mhm.
mentioned, it's just started really. And I
Yeah.
guess those landowners you mentioned, but all the wider community actually, that sort of
Yeah.
feedback to what you've done so far.
Yeah. We organised a hui out in oh, I've forgotten where it was. How terrible of me. Uh we organised a hui earlier this year out in Banks Peninsula, and we fed back to the community, and that was great. I think that now this is on behalf of scientists, actually soil scientists. It's still quite hard to communicate to people the importance of soil because it can seem quite unsexy. Um and So I think sometimes when I've talked to the community about soil and the work I'm doing in the soil, their kind of response is, well, why soil? Why not kind of this invasive plant species that I'm seeing everywhere? Why not the possums that are ruining the forest? Which is completely valid. Those are awful things. um, but sometimes with soil, it's hard to communicate why it's so important, but that's on scientists to work harder at. So yeah, I've had feedback from the community, and it's been good, but we do need to work harder on emphasizing the importance of soil health because it is so important.
Yeah.
And what am I looking forward to most? I'm really excited to, work with the PhD students and really delve into different ways of measuring kind of persistence and stabilization of carbon in soil, not just how much is there, and like kind of testing our different methods and seeing kind of what that kind of data looks like. I'm a bit of a data nerd, So yeah, that's what I'm looking forward to the most.
Well, and kia ora, Kate. Kate, I found who asked you that wonderful question. Kia ora, Kate. Ngā mihi for that. No, well, I honestly just want to reiterate how exciting your work is and actually your wonderful presentation today. And again, Homai Te Pakipaki will give you another virtual acknowledgement to you and your team and wish you well for that. It's wonderful. I think it's so wonderful, this research. It really does show why we need to be doing this research. And kia ora to the third great Rangahau Rising presentation today. Kia ora, Alexa.
Yeah.
Um, so no, it's wonderful research, Alexa, and all the best for that. And we wish you well for that. Any last comments or requests? Otherwise, I will close our presentation today with karakia. But I did also want to promote something else. That's right. Remember that we've got the amazing mahi of Huata and the kupu o te rā, word of the day, and also e mohio rana koe, like did you know? So make sure you've if you haven't been getting those updates through the emails or others, then all the socials, make sure you subscribe so you get those little prods to recognise Aki Aki Aki, our 50th year of Te Reo Māori Language Week. And so I just want to thank everyone here for coming and sharing their whakaaro and their pātai and their questions. Kia ora, thank you. And wishing you all a safe, fabulous week in the meantime. And here's the closing karakia. And again, the app Tuku Tuku, which you can see again how to get the app up there in the chat and join me in our closing karakia today. Ka whakaria te tapu, kia wātia a te ara, kia turuki. Whakatahe ai, he turuki, whakatahe ai, humie hoie tae ki e. And really pleased you could all join us. Ka kite anō. Take care.